The overall goal of this procedure is to surgically isolate, remove, and doubly cannulate individual resistance arterioles for study in an ex vivo setting. This is accomplished by first identifying and surgically isolating a microvessel in an anesthetized animal. After transferring the microvessel to a vessel chamber, the proximal and distal ends of the microvessel are both cannulated. Finally, the microvessel is exposed to pharmacological or physiological challenges and experimental interventions to assess mechanisms underlying function or dysfunction that are intrinsic to the vessel itself. Ultimately, the results show the patterns and contributing mechanisms underlying vascular reactivity in an isolated setting through appropriately targeted stimuli and interventions.
Generally, individuals new to this method will struggle, because this preparation requires the development of not only excellent surgical skills to be performed under a microscope, but also the patience and fine motor control required to doubly cannulate an isolated microvessel with a diameter that is frequently much less than 100 microns. This must be done with an absolute minimum of trauma to the vessel, or the entire procedure will result only in an unresponsive vessel. Demonstrating the procedure will be Joshua Butcher, a graduate student from my laboratory.
Prior to carrying out isolated microvessel preparation, begin by bending previously prepared glass micropipettes to an appropriate configuration by heating them over a butane flame. Then, use fine forceps to break the tips to an appropriate diameter. Place 2 micropipettes in opposition into the pipette holders for the microvessel chamber, orienting them so that the tips are in the same vertical and horizontal plane within the vessel chamber.